Welcome to Bioinfo Bytes, where we decode bioinformatics. Today, we're jumping into a true giant of the field, BLAST. This is the tool that didn't just make research faster, it completely changed the game for modern biology. And we're going to break down exactly how it works.
So let's set the scene. You've just sequenced a brand new protein. But what is it? What does it do? To figure that out, you've got to compare it to, well, pretty much every known sequence out there in these massive global databases. We are talking billions of entries. So how on earth do you find a match in all that noise without waiting forever?
Well, you could try the brute force method, checking your sequence against every single other one, position by position. The problem? That is incredibly slow. Like, it could take you days or even weeks to get one answer. But BLAST gets you there in seconds. And it does this by using a really clever short count.
Okay, so what is BLAST, officially? The name stands for Basic Local Alignment Search Tool. Its whole purpose is to take your sequence and find the best local matches in a database. Notice I said local. It's not looking for a perfect end-to-end match. It's hunting for those crucial regions of really strong similarity. And in the field, we call these matches high-scoring segment pairs, or HSPs for short.
Now, for the really interesting part. How is it so incredibly fast? The secret sauce is a smart bit of computer science. It's a heuristic approach called the word method. The whole idea is, instead of getting bogged down comparing entire long sequences, BLAST just starts by looking for very small identical pieces first.
The entire algorithm is this really elegant three-step process. First, it breaks your query sequence into a list of short words. Second, it scans the whole database looking only for exact matches to those words. And then finally, once it gets a hit, it takes that small match and extends it outwards to build a full alignment. So let's look at each of those steps.
Okay, step 1. Breaking it down. BLAST just chops up your sequence into small overlapping words. For a protein, that's typically 3 amino acids long. But here's the really genius part. It doesn't just look for those exact words. It also creates a list of similar neighboring words. Ones that are biologically very similar and would score high anyway. This makes the search both incredibly fast and really sensitive.
Next up, the scan. Now that it has this optimized list of high-scoring words, BLAST just flies to the database, looking only for exact matches from that list. This is the heart of its speed. It's not trying to align anything big yet. It's just looking for these tiny, promising starting points. We call them seeds.
And that brings us to the final step, extension. As soon as BLAST finds one of those word matches, a seed, it starts to build the alignment outwards from there, in both directions. It adds one letter at a time and keeps going as long as the similarity score improves. This whole process is what creates the final result, a high-scoring local alignment, RHSP.
Okay, so BLAST ran its search and it found some matches. But how do you tell what's a meaningful biological connection versus what's just random noise? Let's take a look at how you actually make sense of the results.
When you're looking at your results, you really want to focus on 2 key numbers. First is the bit score. It's a score that measures how good the alignment is. The rule is simple. Higher is better. The second number, and I'd argue it's even more important, is the E value. That stands for expect value. It tells you how many matches with a similar score you'd expect to find just by pure chance. So for the E value, the crucial takeaway is you want a low number. The lower, the better. Anything under 0.05 is generally a pretty good sign that your match is real.
And this is exactly what a real BLAST output looks like. You can see the matches are all ranked. The best hits, the ones you want to look at first, are always right at the top, with those high bit scores and those really tiny significant E values.
One last thing, and this is super important. BLAST isn't just one program. It's actually a whole suite of different tools, and choosing the right one for your specific question is absolutely essential to getting a good answer. So let's quickly run through the main types.
You can think of this as your quick start guide. If you have a protein and you're searching a protein database, you use blastp. For DNA against a DNA database, it's blastn. Now it gets interesting. Say you have DNA but you want to know what protein it might make, you use blastx. It translates your DNA for you and searches a protein database. The reverse of that is tblastn, taking your protein and searching a translated DNA database. And finally, there's tblastx, which translates everything both ways. That's the one you use to find really distant evolutionary relatives.
And that's really the heart of it. By making a smart trade-off, giving up guaranteed perfect accuracy for just incredible speed, BLAST unlocked our ability to navigate this massive, growing ocean of biological data.
It's not just a tool, it's the fundamental search engine that so much of modern biology is built on. It's fast, powerful, and absolutely essential.
Thanks for joining us for this explainer. If you want more bioinformatics decoded, make sure you subscribe. Join us next time as we dive into global sequence alignment with the Needleman-Wunsch algorithm, right here on Bioinfo Bytes.